The Global Consortium just had its meeting, and Dr. Prakash is on to another meeting concerning genetics. Now, this is my disclosure. Trust me, I have no interest in any of the manufacturers of genes. Genetics now is of high interest to everybody. It first started for ophthalmology in the Journal of the Research, ARVO, and went into ophthalmic genetics, and it then got into the general literature for ophthalmologists. The archives of ophthalmology have centered it, and even the publication of the American Journal of Ophthalmology of the Academy. Then it went into the consumers, Evolution, Newsweek magazine, Time magazine. So we're at the point where our patients are knowledgeable, and we've got to become knowledgeable to catch up with them. After all, we are their doctor. This is a takeoff of what Mahatma Gandhi said. Okay, we have to look at ourselves. A little baby, what's the future going to be? Illnesses, sickness, evolution, intelligence, physical attributes, all that's genetic. Parents want to know. And if necessary, parents want to modify the genetic makeup so that their child will be healthy and succeed. We'll be going into that. Here's the genetic code. Now, we know what the codes are for diabetes, breast cancer, autism, Alzheimer's disease, obesity. It's all there. Can we decode it? Can we change it? Stay with us, you'll find out. So this is what genetic testing can do. It can predict a medical condition to a certain extent and diagnose certain genetic disorders and what foods to avoid. But there are limitations. We are disappointed because there are variations that it detects little of genetic links to most diseases. Why? Because a lot of the diseases are polygenic. There are several genes that are responsible for it. And of course, they're multifactorial, environment, hormones, etc. A lot of interference with the genes. So what do we want to do genetic-wise? Well, we want to be able to tailor drug therapy to an individual's uniqueness based upon their single nucleotide polymorphisms. Gene therapy, if possible, and the antisense drugs and DNA vaccines, because we know that viruses can penetrate the cells and carry with it the altering genes, especially if needed. And then we have the recombinant drugs that could alter the genes, and you'll see that in a minute. If I push the right button, here's the antisense, where it can interfere with the mitochondrial RNA translations into proteins. And we have some drugs and classes of drugs that do that. So let's get down to the basics. We all know about the purines, adenine and guanine, and the pyrimidines, thymine and cytosine. That's the basis of all life, not only animal life, but also plant life. These are the basics. So what do we do with it? J. Craig Venter, I'll bring up several times, change the DNA software, change the species. And here it is. He is converting the genetic code into a digital code. And he spoke at the ARVO meeting many years ago. He has many places. He's listed as Boston, but his main research labs are just down the street from where I live in La Jolla, California, on the campus of University of California, San Diego. And what he always talks about is changing Mycoplasma mycotes, a large colony, into the capreolum cell, and where you could change, engineer bacteria and put yeast vectors into bacterial genomes and the transformations that take place. A lot of this has been printed in the New England Journal of Medicine about changing gene lines and the use of the associated adenoviruses. How it affects us. We have scientists regenerate retinal cells, and vision sciences are also working on retinal cells in the eyes of adult mammals. Major article in New England Journal of Medicine several years ago, Genomics and the Eye. The reference is down below. Gene therapy, vectors, as I mentioned, they were usually viruses that had been genetically altered to carry the normal human DNA to deliver therapeutic genes to a patient's target. And we can manipulate the viral genome to remove a disease-causing gene and put in therapeutic ones. This is one used for trabecular meshwork, just the various carriers. And the ones that we usually look at for ophthalmology would be AAV2 and 9. AAV is the gene delivery tool. Single-stranded DNA genomes that changed into transgenes and aimed at the nucleus and can cut out the disease gene and with transgene put in the new genes. Here's how it works. We could see it's on the surface, penetrates the surface, it's carried down below, and finally enters the nucleus and makes the appropriate protein that you want. For ophthalmology, yes, this has been injected, but unfortunately, when you inject in the vitreous, only 10% hits the retina. If you do it underneath the retina, you have to stay away from the fovea because it can thin the fovea. And we're looking at treatments that are placed in the periphery of the retina and can then migrate and change the cells of the fovea. So here are some of the diseases of the outer retina that you're familiar with: AMD, retinitis pigmentosa, et cetera, et cetera, et cetera. These are the ones that we want to treat. And once again, J. Craig Ventnor changed the DNA software, changed the species. So in molecular therapeutics, one of the things that we want to do is reach a point of knowledge where a certain patient's genetics allows for development of specific therapeutic strategies, which I mentioned before. And with that, we have the new field of CRISPR. That means clustered, regularly interspaced, short palindromic repeats. What does that mean? Well, it's a double-stranded sequence of DNA, but the order of the nucleotides are the opposite on each strand. And each repetition is then followed by a short segment of spacers, and then we can cut with the CAS system. Think of CAS, which is the CRISPR-associated system. Think of it as a pair of scissors. And how is that delivered? Well, it's delivered by RNA. It's delivered by the synthetic guide RNA, and with that guide, it can cut out the DNA that you don't want and put in a new one. That's simplified. And with that, we could even create double-stranded breaks. In the old days, there was zinc finger nucleases that did it, and transcription activated like effector nucleases, but this required a lot of work. With CRISPR, it could be done quickly and on short RNA sequences. And in biomedicine, this has created mutations in human pluripotent stem cells, novel antimicrobial therapy, and individual cancer treatments. It could delete a gene that could interfere with the immune system's ability to fight cancer. But it gets worse, maybe better. In human embryos, we have controversy because it was tested on non-viable embryos, altered the DNA to correct mutations. So in this case, social Darwinism through gene modification. And this was also highlighted just 2 weeks ago in the New England Journal of Medicine, where they go into a lot of detail. It's a prime time for genome editing. And you can see, again, the CRISPR-Cas9. January 30th, you can pull it up. And it gets worse. It gets worse. Here's the Chinese scientist, He Jianjing, at the Second International Summit for Human Genome Editing. He then disappeared. And it turned out that he was sentenced to jail for 3 years because of his work on gene-edited children. He shocked the scientific committee when he announced in Hong Kong that he had genetically altered and modified the embryos of twin sisters that had been exposed to HIV through their father, and he was able to create the gene to knock out the receptor site in their DNA to HIV, modified them, this gene called CCR5. I want to go into, I'll take a few more minutes, that this is the RPE65 gene dystrophies and what's being done. Leber congenital amaurosis, retinitis pigmentosa, night blindness, and we can see the various genes affected. And in comes Luxturna for Retagene, developed by Spark Pharmaceuticals. And what this does is it can change the RPE65C DNA. And it's only right now used for Leber congenital amaurosis and retinitis pigmentosa. But look at the price, close to $1 million American dollars, not rupees, dollars. And it's one-time treatment. It's broken into two, one for each eye. I'll quickly go into epigenetics, which is the study of hereditary changes in gene expression by mechanism other than the changes in DNA. And so this was brought out by Jean-Baptiste Lamarck. And here it is with the giraffes, that they were able to develop tall necks because of the necessity to eat the foliage high up, where it was of value. These are the molecules that attach to the DNA, methyl and acetyl groups. And by altering that, you could alter, and it could carry forward into posterity, the appearance and the work. So let's go from genetics to stem cells. Tissue engineering, and this is the replacement of defective tissue with cultured stem cells, the pluripotent, and this is an unfertilized blastocyst, and this is a source of stem cells which is used to a certain extent. And then we could take a person's own tissue, dermatocytes, and make them into pluripotent. And this is just a corneal orb that's created from those stem cells from the pluripotent. Here, this is the transcription factors where a person's own fibroblasts are changed into potential stem cells, and then other tissues can be developed. And even the human eye, here's the references. I appreciate you watching. Come to the future with me, and we're off. Our next speaker is Dr. John Prakash from the National Eye Institute in Bethesda, Maryland.